Okay, everybody. So this step is preparing the media for growing the bacterial culture. So Yonghua is going to demonstrate how to make a small LB media, which we will then autoclave and then get it ready to basically grow cultures with. Go ahead, Yonghua.
So this is the LB broth, and as you know, we can tear 4 container. Measure proper amount of LB. Then put in. It's a small culture, so we're using 250 ml flask. And we'll use appropriate water. Once you make it, you don't have to actually mix it. Once it get in the autoclave with the high temperature and pressure, it'll just all mix itself. Then you can put aluminum foil on top. You don't have to seal very tight. If you do it, because of pressure, it just the cover will pop open. So you want to kind of loose. Then this is tape for whether you autoclave or not. It will tell you. So if you see, you probably cannot see. There is a line. Once you autoclave, it's showing like a black stripe. So put on top of it. So that's how you make LB media.
Right. So the demonstration is only for the smaller media, but you can make the bigger ones the same way. Use larger flask, right? So everything is proportionally bigger, but it's the same deal, and we'll just demonstrate only with a smaller one. Okay. Okay, now we're ready for autoclave, and this big machine is the autoclave. So Yonghua has placed our media in there, okay, with the tray. So now she's going to operate. This part you don't have to learn how to do because you probably don't have to do it, but we want to show you how this is usually done. An autoclave is to sterilize the sample by high temperature and high pressure. Okay.
So once you're done, autoclave option, there are 3 in this machine. Usually dry one for solid and liquid. Anything wet, you choose this option. The other one is waste. So now LB media is liquid, right? So we'll choose option 2 of liquid. Once you push it, this machine itself, we set up all pressure and temperature and time and cooling temperature. So what you need to is, right now, the jacket was full, so it's just emptying out. So once all jacket is empty out, you can see there's a big button, start. It's very easy to operate. You just need to push the button. Then machine itself automatically process until done. It will take for this machine 45 minutes, so we'll check out after 45 minutes.
All right, so the autoclave is finished. Yonghua is going to open.
The push button, open lower button. This has been a while, so it's not really hot. But if you just right after done, you should wear gloves.
Alright, so you can tell the tape has turned color. Now it's black. That means the autoclave has finished successfully. The thing is sterilized and ready to go. Okay, until once it's cooled down. All right. Alright everybody, so this is experiment number 9. We're doing protein expression, and so for part A of this experiment, it's transformation. of the plasmid that we cloned and verified in experiment number 8. So we know it's correct, and we have finished transforming that plasmid into E. coli cells for expression, right? So this today we are starting from part B, because part A, the transformation part, you've seen already in experiment number 7, so we're not going to repeat that. But we will start from basically part B, which is where we already have the colonies from transformation. You have the plate here with colonies on there, so Yonghua is going to then pick one of the colonies to inoculate the media that we prepared, small media, LB, and then let it grow. So again, Yonghua couldn't talk much because again it's sterile. So Yonghua is going to pick a single colony with a pipette and very carefully place this colony into the LB media. Right? It's now sterile after the autoclave, and it has been cooled so that it's ready for inoculation. And she's also going to add ampicillin, the thing that's going to select for our bacteria that contains our plasmid. Okay, so this is everything needed. Alright, so this small culture is now ready to go into the shaker. Right. Okay, so it's the same shaker, and it's set at 37 degrees Celsius. It will go overnight, and after the overnight shaking, the culture will hopefully become dense, meaning that the bacteria has amplified enough and ready for our next step. Okay, everybody, so it's the next day. Sorry, we're pretending it's the next day because we don't have the time. So this is the small culture that we placed in the shaker earlier. It's supposed to go overnight and then grow to certain density, right? It should be really cloudy and dense. However, we didn't have the time, so it looks clear now, but it shouldn't be that way. It should be really dense, having a lot of bacteria in there. That means the bug has replicated, reproduced, amplified enough so that it's in the right density to be used for the next step, which is to inoculate this bigger media. Okay, it's similarly autoclaved, ready to go, and when you have certain volume of dense bacterial culture going into there, you will inoculate the larger media, which will allow the bacteria to grow even more. Okay, so then when they produce protein, we'll have a lot of protein produced. Okay, so that's the thing, and basically we will be using this not really dense culture to demonstrate how that's done. All right, go ahead, Yuhan.
So, like Dr. just said, everything is sterile. So you just needed to dump small flask to big flask, and you put, of course, you still have to put your ampicillin.
Right, the antibiotic in there.
Like a proper amount of volume that required. So everything is ready to go bigger shaker.
Okay. So that's the big shaker. Yuhua is now placing the flask into one of the holders. And she'll set the temperature to 37. Okay, and then this will be basically shaking in there for a few hours. We'll be monitoring the OD, which is the density of the culture, right? When it reaches certain density, we will then perform the next step. All right, 37 degrees. Okay, so the culture has been there for a few hours. It's not really ready yet, according to the UV, but we're gonna take a gel sample for right now, so that we have uh, a way to know how much protein or whether a protein of interest is being expressed already. So Yunhua simply took some of the culture and placed into a clean tube, and this tube, can I show it? So this tube containing the culture will be spun down, and we'll discard the supernatant, keep the pellet at the bottom, and then we'll add the gel dye and everything else to make gel sample out of that pellet. Okay, and that pellet, when you run it on the SCS gel, will tell you all the proteins that you have in the sample, and we'll compare this sample with the sample which we'll prepare later after the protein has expressed. We'll compare these two side by side to then basically visualize whether our protein of interest is being produced and roughly how much is produced. Okay. Okay, so we are now checking the OD. It's almost there, so hopefully we are aiming for OD between 0.6 and 0.8.
Yeah, we have blank with water.
So we're again measuring the OD at 600 nanometer wavelength.
So we can just take from flask.
Right, we took the culture out, and then Yunhua is retrieving some sample.
So right now it's right OD over 0.6.
Right, so now the culture is ready to be induced with IPTG.
So this is our IPTG already made.
So as you probably have learned in the lecture, IPTG is inducer, right, to induce protein expression. So at this point, when the E. coli cell has grown to a certain density, it's ready to be induced. So the protein will start to be expressed from this point on, and hopefully give us a decent yield in the end. So this culture is now going to return to the shaker. And the growth will be continued at 37 degrees for a few more hours, and this is the time again when the proteins will be produced, hopefully in large amounts, so that we have more proteins to work with for our next experiment. All right. Okay, everybody, so the growth has finished. We're ready to spin down the culture, but before we do that, we're going to save a final gel sample to analyze our expression results. So Hua has taken some of the culture and placed it into a clean tube. Right, so that is the thing we'll be basically spinning down, and then save the pellet, discard the supernatant, and that pellet will be prepared into a gel sample, which we'll analyze later. Okay. All right.
So we are spin down. Before we spin down, we have to make exactly same amount of volume of container. So we are using scale. for precision.
Yeah, so again, the key is to make sure that the two bottles will have the same weight, the exact weight, right? So when they go into the centrifuge, they don't cause any trouble. Okay? When they're balanced, machine is going to run smoothly. If they're not balanced, they will damage the machine. So it's very important what Yunhua is doing to basically make sure the two bottles have the same weight, the exact same weight.
Yeah, similar enough.
All right, good. So we'll then close them tightly and move to the centrifuge to spin it down. Okay, so here is the centrifuge, right, and that's right the rotor, and that's where the bottle is going to go. Have to make sure the lid is put on tightly and properly.
And we choose, there are lots of different motors, so we choose our motor, which is 55. And you can choose your RPM and time. Temperature will be 4 degrees. So once everything is set, you can just push the button start.
Right. And that's basically the last step of the experiment, right? So after the spinning, after the 50 minutes, we'll again dump out the supernatant, and the pellet at the bottom will be recovered, will be saved for our next experiment. Okay.